The next experiment is on a temporary preparation of a tissue. That is squamous epithelium. Squamous epithelium is obtained from two different sources. The cheek is scraped with a blade. A slide is taken, and to the slide, a small drop of egg albumin is on the slide. It's taken a very small drop of albumin, egg albumin. The albumin is rubbed on the surface of the slide. It's rubbed on the surface of the slide like this. Okay. A drop of water or two is taken on the slide. A drop of water or two. First of all, albumin is rubbed on the surface of the slide. Then a drop of or two water is put in the middle of the slide. Then the slide is held like this. It's held like this. With the help of a blade, the cheek is scraped gently. Cheek. Remember, the cheek is scraped gently so that the scrapings will fall on the slide. The winter season has begun. Now you can get easily these scrapings, cheek scrapings. You scrap it gently. Take care that there is no injury to the cheek. Then the scrapings, the entire slide is air dried. Put on air dried. It's dried. Now its scrapings adhere onto the slide. They will not be displaced any longer from the slide, from their position on the slide. They stick or adhere onto the surface of the slide. Then the slide needs to be stained with a suitable stain solution. What is that? We use either of the two stain solutions. Hematoxylin, aqueous hematoxylin stain. We put a drop or two of aqueous hematoxylin stain here. Or eosin stain, aqueous hematoxylin stain or eosin stain, whatever the case may be. Just allow 5 minutes transit time. Let the stain be there for 5 minutes. See that the stain doesn't dry up completely. It doesn't dry up completely. If it dries up, or if it is on the verge of drying up, you add 1 to 2 more drops of the stain. Okay. After 5 minutes, waiting for 5 minutes, you wash the slide thoroughly with water. Wash totally. Slide cover. Please wash the slide. Raise the slide gently. Okay, we raise the slide gently. It's raised. Excess of stain is removed. Then allow it to dry up. It dries up. When it dries up, you put a drop of glycerin at the center. And on the glycerin drop, you put a cover glass. Seeing that no air bubble enters into the glycerin droplet. Now the slide is ready for observation, microscopic observation. With the help of a compound microscope, you observe the slide at 10 into 10 magnification, at 10 into 10 magnification. 10 into 10 magnification means, or it refers to, the eyepiece must be 10 and the objective lens must be 10. So 10 into 10 magnification. So whenever magnification is computed, both the lenses are taken into account, both eyepiece and objective lens. So they are multiplied together. 10 to 10 magnification means whatever is the dimension of the material, if you observe it under a microscope, it will look 100 times, 10 to 10, 100 times more than that of its observation with the naked eye. Right? So, the slide is observed at 10 to 10 magnification in a compound microscope, and look at these cells. These are the cells. Upper view of this squamous epithelium. If we take a profile, side profile, the cells will look like this. The cells are very closely fitted, tightly fitted onto each other without any intercellular space or any intercellular substance. They are flat cells. Each cell has a nucleus at the center, consequently bulging the cell at the center and tapering the cell at the sides or at the periphery, such that the cell looks spindle shaped like this. As you can see here, this cell is spindle shaped. At the center there is a nucleus. Due to the presence of the nucleus, this cell is bulging at the center. And at the peripheral part, the cytoplasm has narrowed down. And this is a look, this is another look. This look is from the upper side and this look is from the lateral side, side view. This is upper view and this is side view. Squamous epithelium. It's very easy to get squamous epithelium from the scraping, thick scraping. Another way of getting another easy source of squamous epithelium is that is forbidden. I shouldn't. Anyway, I will tell you, the choice is yours. You take a toad, Bufo melanostetus, toad. Put a toad in a trough of water for some time. Put a toad in a trough of water for some time. You will notice that the toad discards skin flax. These skin flax are the source of squamous epithelial cells. But why I am referring it to as a forbidden material? Because the toad is a live toad. You cannot sacrifice any animal for the cause of mankind. Nowadays, you cannot sacrifice any animal. There are laws which forbid the sacrifice. It's a kind of torture. You put the toad in a trough of fresh water, it will discard flakes, remove the flakes. For this petty experiment, you cannot sacrifice the life of a toad. Therefore, it's forbidden. Anyway, so we have an alternate source, chick scraping cells. That's about squamous epithelium. Next, we move on to striated muscle fiber or skeletal muscle fiber or striped muscle fiber, as you may call it. Three synonymous terms. You make a temporary microscopic preparation of the skeletal muscle fiber. What is that? What is a skeletal muscle? A skeletal muscle is a muscle that is attached to the endoskeletal element of the body of a vertebrate. Um, to femur, to humerus, to vertebrae, to girdles, to skull bones. All these are skeletal elements. To any cartilage also, skeletal muscle is attached to endoskeletal elements. In order to prepare a skeletal muscle fiber microscopic preparation, you require or you need a preserved skeletal muscle. You remove the skeletal muscle, then preserve it in formaldehyde solution for a longer period of time. Remove a piece of skeletal muscle on the day of preparation. On the day of preparation, use the preserved skeletal muscle, remove a small piece of skeletal muscle from it, put it in water for washing, because as long as formaldehyde solution is there in the muscle fibers, the fiber cannot be stained with a stain solution. Therefore, you have to remove the formaldehyde solution. You put it in water, wash it, then tease it. Teasing means you hold two needles. You have taken the skeletal muscle on a slide. Now you will tease it. Teasing means division, subdivision, again division, subdivision, subdivide until the skeletal muscle turns into the minutest fiber. You take two needles like this. Put it here. You take no muscle is there. So think that muscle is here. No muscle is there. Anyway, so you tease it, divide it into two fragments. Each fragment is then divided into two fragments. Again, two fragments. Go on dividing and subdividing like this until you get the minutest fiber. Until you get a fiber such that you cannot see it. That is how the skeletal muscle fiber is prepared. Ground substance. It is prepared, okay. Through teasing, discard everything except 2 to 3 fibers. Then put 2 to 3 drops of aqueous hematoxylin stain solution. It's put. Wait for 5 minutes. Wash it. Then put a cover glass. As usual, whatever is done. You put a drop of glycerin at the center. Put a cover glass on it. Then go for microscopic observation. If you using a compound microscope, at 10 into 40, it's not 10 into 40. 10 into 10, first of all, you need to see it at 10 into 10 magnification. At 10 into 10 magnification, this slide will look like this. A. So, what is there? The fiber, a muscle fiber, is a multinucleate structure. First, it's a multinucleate structure. There will be many nuclei in the common mass of cytoplasm, that is sarcoplasm. This multinucleate condition is known as a syncytial condition. So, a skeletal muscle fiber is a syncytium. That is, remember, don't ever forget, a syncytium is a condition where a common mass of cytoplasm has several nuclei. it's a multinucleate condition. This thing applies to skeletal muscle fiber. Okay. So this fiber is said to have alternating dark and light bands. Alternating dark and light bands. The dark bands are called anisotropic bands. The light bands are called isotropic bands. So anisotropic or A band, isotropic I band. These are alphabetically named like this. I isotropic, A anisotropic bands. Everything is level here. And if we magnify it a little more, this is the look. The light bands, this is clearly visible. You need not have to take the help of a high resolution microscope. In a compound microscope, in your laboratory microscope, you can resolve this kind of a structure. 10 to 40 magnification, you can resolve this kind of a structure. So there will be light and dark bands alternating with each other, and the light and dark bands of one muscle fiber will coincide or synchronize with the light and dark bands of an adjacent muscle fiber. Like this, all the light and dark bands of all the muscle fibers present together will synchronize or will coincide with each other, thus giving us a striped appearance. Sorry. Okay, this is the striped appearance. Isotropic band and anisotropic band. This is a simple preparation, but here the technique, real technique, lies in the teasing process and removal of hematoxylin. Removal of formaldehyde solution, the preservative solution that is present in the muscle fiber. So, muscle consists of several. Actually, structurally, I didn't tell you the structure because we are concerned with the muscle fiber. But as I understand now, the structure is quite necessary for a student. A muscle consists of several bundles of muscle fibers. There will be one bundle of muscle fiber here, another bundle here, another bundle here. So many or several bundles of muscle fibers are present side by side in a muscle, skeletal muscle. Each muscle fiber bundle has several muscle fibers. Each muscle fiber has myofibrils. Each myofibril in turn has myofilaments. The myofilaments are protein filaments. There are two types of protein filaments: actin or thin protein filaments, myosin or thick protein filaments. The protein filaments of one fiber and its adjacent fiber are synchronously arranged such that this kind of an appearance arises. Look at the synchronity. This is a muscle fiber, this is another muscle fiber, this is here another three adjacent muscle fibers. Synchronously arranged. The myofilaments, protein filaments, truly give or impart this kind of a striped appearance. Because there are A bands, I bands alternating with each other. This is how we prepare, make it preparation. Since we are concerned with higher secondary classes, we are trying to make the higher secondary students to understand how we don't go ahead of this. They will put a full stop. Okay. So that's all about tissues. We don't have any more tissue to discuss.